Late July, Almeria, southern Spain, and the pepper plants inside Carlos Moreno's greenhouse were disappearing beneath a living gray crust. 2 million aphids coating the undersides of leaves so densely that the green had simply ceased to exist, row after collapsing row. Carlos is a third-generation farmer. His grandfather planted the first pepper rows on this land, and Carlos has spent his life learning every weather pattern, every soil shift, every pest this region throws at a crop. And still, in that summer, the infestation was winning, slowly and completely, and without mercy. He had already run through 3 separate synthetic insecticide regimens across 11 weeks, cycling from one chemical formula to the next, and the aphid population absorbed every application and kept expanding, a colony that had effectively become immune to the only tools in the cabinet. I'm Dr. Raymond Finch. I spent 40 years in the field watching ecosystems break down and watching them repair themselves when the missing piece was finally returned to the equation. What happened in that greenhouse is not a gardening story. It is a physics lesson about predation, territory, and population collapse. Now, think about what that means for a moment. Moreno was staring at a harvest valued at over 80,000 euros, and the intervention that would ultimately save every row of it cost him less than 300 euros in total. A number so small against the loss that it barely registers until you understand what was inside those boxes. An agrobiologist from the local cooperative proposed something that sounded, to Moreno's practical ear, almost absurd. Release 10,000 Coccinella septempunctata, the common 7-spot ladybug, directly onto the infected crop, and simply observe what a predator does when you hand it back its territory. Here is where it gets interesting. A single adult ladybug consumes up to 50 aphids per day, which means 10,000 individuals represent a predation force capable of eliminating 500,000 aphids inside a single 24-hour cycle. A biological pressure no-spray schedule in that greenhouse had come anywhere close to matching. Within 72 hours of release, field technician Daria Solano, stationed at the cooperative's monitoring post in Almeria in the summer of 2019, documented something she had not predicted and had not seen modeled in her training data. The aphid population across the first 6 treated rows had not merely declined. It had collapsed. The gray crust was dissolving row by row, as though the plants themselves were exhaling for the first time in weeks, stems straightening, leaves uncurling, the green returning to surfaces that had been buried under living weight for nearly 3 months. You have to understand how rare it is to watch a system pivot that fast. What you are probably asking right now, and it is exactly the right question, is whether this was a single lucky release into a single desperate greenhouse, or whether the underlying mechanism Solano documented that July holds across different crops, different infestations, and different scales of infestation entirely. Stay with this, because the field reports from the next 18 months are what change everything. Was this a one-time miracle, or a repeatable ecological law? The leaf surface told you nothing. That is the brutal, expensive truth that Daria Solano spent 3 seasons trying to put into numbers that growers and agricultural authorities would actually believe. You look at a row of peppers in full summer growth and you see health. The green is convincing, almost theatrical in how ordinary it looks. What you cannot see is the underside of every leaf, where colonies dense enough to drain a plant's vascular fluid within 10 days are already at work. This is the part that made me stop and reread the field notes. Solano's baseline surveys, conducted from 2016 through 2018 across 4 distinct growing regions in southeastern Spain, did not reveal a pest problem. They revealed a system's failure that had been running quietly for years beneath the green. In the Campo de Dalias coastal plain, colony density had reached 800 individuals per leaf cluster by mid-season, enough to cut chlorophyll production by nearly 30% before a single yellow patch appeared on the surface. Across the Murcia pepper belt, 3 consecutive seasons of imidacloprid treatment had produced a measurable 22% rise in resistance. The chemistry was losing while the invoices kept arriving. In the Valencia corridor, growers on a 9-day spray rotation were still recording aphid-attributed losses, averaging 18% of total yield in post-harvest audits. And along the Almeria coastal strip itself, where roughly 30,000 hectares of protected cultivation sat in near-continuous production, the regional authorities' own 2019 data showed aphid management had become the largest input cost for pepper and tomato producers, averaging €1,400 per hectare per season. 800 individuals per leaf cluster. Hold that number for a moment, because that is the density at which a plant begins losing its ability to photosynthesize before any outward sign of distress reaches the surface, and before any grower has cause to call anyone. At €1,400 per hectare per season, aphid management was not a line item. It was the line item, the cost that shaped every other decision a grower in that corridor made, from planting density to harvest timing to whether they could absorb one more bad rotation. The resistance data from Murcia is what stopped Solano's team completely. A 22% measurable increase in imidacloprid resistance across 3 seasons meant the primary chemical tool growers had been given was not controlling the infestation. It was selecting for the survivors who could tolerate it. You are probably forming the question that every grower in that region eventually formed. If the chemistry was failing and the audits confirmed it, why did the spray schedules keep running? That answer is coming, and it is not a comfortable one, so stay in your seat. What Solano's 3-season baseline actually documented was a 3-part collapse hiding beneath normal-looking rows, a resistance curve accelerating faster than new chemistries could enter the market, a yield loss figure that post-harvest audits kept confirming, but mid-season visual surveys kept missing, and an input cost that had become structurally embedded in how the entire corridor operated. What looked like a managed system was a system in slow chemical surrender. The green rows were not evidence of control. They were a delay, and the 2019 season in Almeria was where the delay ran out. And that is the condition, that precise, documented moment of chemical exhaustion, into which 10,000 predators were introduced, not as a hopeful experiment, but as a last calculation by people who had run out of other options and finally read what the data had been saying for 3 years. So 10,000 predators entered a greenhouse in Almeria in July 2019, and the calculation that put them there was not optimism. It was arithmetic born from 11 weeks of documented failure that had exhausted every other option on the table. Carlos Marino's operation ran 3 separate insecticide applications across those 11 weeks, and every time the colony counts dropped, the rebound came in 8 days. A biological clock no spray schedule could outrun, repeating itself with the patience of something that had learned the timing. The combined cost of that program, product, labor, re-entry downtime across 4 workers, exceeded €4,800, and the aphid population at week 11 sat within 15% of where it had started, which is not a management result, it is a subscription. Here is where the data stops being data and becomes something else entirely. Because in 2017, the Regional Growers Association had formally advised its members against predatory insect releases, and that language, as Daria Solano documented in her 2021 published field report, had been lifted nearly word for word from a briefing co-authored by a pesticide distribution consortium. That 2017 advisory carried real weight across the province. It shaped purchasing decisions, it shaped what agronomists recommended, and it held for 2 full growing cycles before the 2019 collapse made the argument impossible to sustain any longer. €280. That was the purchase cost for the 10,000 ladybugs. No reapplication schedule, no re-entry interval, number 1 40-hour labor rotation, just a box of living predators and a door left open long enough to let them disperse. Within 4 hours of release, measurable predation had begun, a single adult ladybug consuming up to 50 aphids a day, and with 10,000 working the same rows simultaneously, the arithmetic was no longer theoretical. You are probably wondering right now whether a collapse that fast, documented by Daria Solano across the first 6 treated rows within 72 hours, was a data error or a genuinely repeatable field result, and that question is exactly the right one to hold, because the answer determines whether everything that followed in Almeria was an exception or a proof. They said it could not perform at commercial scale. What Solano's July 2019 observation opened, an aphid colony collapse that had not appeared in her training data, that no spray rotation in the province had replicated across 3 consecutive growing cycles, was not a curiosity. It was a controlled, documented result that the next act will follow all the way to its conclusion. What Solano's July 2019 observation opened was not a single event. It was a sequence, and sequences have physics. The first movement happened inside 4 hours of release. 10,000 Coccinella septempunctata dispersed laterally, spreading low and wide across the canopy rather than climbing vertically, moving not toward the nearest feeding site, but toward the heaviest aphid density in the greenhouse. Solano called it purposeful in her day, one field notes, and that word mattered, because it meant the insects were not scattering randomly the way released populations often do in unfamiliar enclosed environments. They were navigating. They were reading the air. The chemosensory precision of what she observed was something no spray application had ever replicated, because a spray cannot choose where it goes. By hour 72, aphid populations on the first 6 treated rows had fallen by 68%. And here is the part that made me stop and reread the field notes. That drop was not purely from predation. Alarm pheromones released by consumed aphids caused the surviving individuals to disperse, fracturing the structural density of the infestation faster than any beetle jaw could have managed alone. The cascade deepened on day 4, and this is where the biology stopped behaving like a controlled trial and started behaving like a system. Parasitic wasps, Aphidius colemanni, began arriving in the greenhouse canopy entirely uninvited, drawn by the same alarm chemistry the dispersing aphids had been broadcasting into the enclosed air. Carlos Moreno had not purchased the wasps. He had not scheduled them. They were already present in the surrounding Almeria landscape, and the alarm chemistry pulled them in. A second predation layer that cost exactly €0 and contributed measurable pressure that no protocol had anticipated or budgeted for. The cascade had recruited its own reinforcements. Now, think about what that means for a moment. A farmer paying €280 for a biological release inadvertently activated a landscape-level predation response that no synthetic rotation had ever managed to trigger, because a synthetic rotation kills the chemistry along with the pest and leaves the air silent. You may be wondering whether the plants themselves showed any measurable response. And if that thought is forming, hold it, because the answer is coming and it changes the scale of what this 9-day period actually proved. The plants were not passive witnesses to the predation happening above their vascular tissue. By day 6, new lateral shoot growth had appeared on 3 of the 6 treated rows, a physiological recovery signal that cannot occur until a plant has sustained at least 4 consecutive days of reduced vascular stress. The plants were not recovering because someone intervened. Further, they were recovering because something had finally stopped. By day 9, Solano ran her grid count protocol across 200 individual leaf samples and arrived at a figure she counted 3 separate times before she trusted it: a 94% reduction from the pre-release aphid baseline. Not managed, not suppressed. Reduced, measurably and verifiably, to near absence. The 94% figure was not the product of 10,000 beetles eating 10,000 individual insects. It was a cascade. Predation fractured structure, structure loss erased reproductive momentum, and the infestation that had resisted 11 weeks of chemical application unraveled in 9 days because the biological chain, once set in motion, fed itself. What happened next? How Solano's field report moved from a cooperative monitoring post in Almeria into the hands of agrobiologists who had spent careers defending the chemical model is a story about what happens when a number that large cannot be argued away. The protocol was clear: stay within the central cultivation zone, concentrate in the 6 designated rows, and do not wander. The Ladybugs had not read it. By day 3, Daria Solano's recount surveys across adjacent rows told her that roughly 1,800 individuals had drifted out of the treatment zone entirely, moving into the untreated tomato section that nobody had flagged for any kind of pressure whatsoever. That number, 1,800 insects self-redistributing without instruction, without a coordinator, without a map, represented 18% of the entire released population, making a collective navigational decision that no model had anticipated. The grower, Moreno, made 2 calls to the cooperative in the first 48 hours, reporting that the insects were out of control. He was not wrong in what he saw. He was only wrong in what he concluded from it, which is a distinction worth holding on to. When Solano physically walked the tomato section on day 4, She found an early-stage colony already established across 12 plants. A colony too young to have crossed the visible reporting threshold, invisible to the grower, invisible to every standard protocol, but apparently not invisible to an insect navigating by scent gradient. The unintended outcomes divided cleanly into two categories. The first, a preemptive interception that Solano calculated would have reached reportable infestation density within 11 to 14 days had it gone untouched. A secondary outbreak nobody budgeted for was stopped by insects nobody directed. The second, a genuine regulatory breach nobody had planned for either. Now, think about what that means for a moment. The insects had effectively run a preemptive strike on a colony that did not yet exist in any official record. They found the problem before the humans with clipboards did, and the humans with clipboards had been walking those same rows every week. By day 12, approximately 600 ladybugs had moved through a ventilation gap and into an adjacent greenhouse owned by a grower who had given no consent to the original release program. That greenhouse grew sweet pepper, and under regional phytosanitary protocol, any introduced biological agent required prior documentation before entry. The cooperative spent 3 weeks inside an administrative process resolving what the paperwork called a cross-contamination event. Solano, in her written report, described it as a paperwork problem caused by an animal that cannot read property lines, which is precise and correct, and also exactly the kind of friction that gets biological control programs shelved before they scale. You are probably wondering whether this administrative friction was enough to stop what Solano's data had started, whether a 3-week paperwork delay was enough to convince the agrobiologists who had spent careers defending the chemical model to look the other way and walk back to what they already knew. That answer is coming, and it is not the one you are expecting. The ladybugs had exceeded every spatial model not out of defiance, but out of accuracy. They were following a food gradient, which turns out to be a more reliable navigation system than any protocol a human committee has ever written. What the scientists did with that fact next is where this story takes its final, unexpected turn. Daria Solano's final field note from the Almeria release, published in 2021, closed with a sentence that has stayed with me since I first read it. The system did not need a new tool. It needed its old one returned. That single sentence is the whole architecture of what happened in that greenhouse. Not a novelty, not a fortunate accident, but a restoration of a predator-prey relationship that had been operating on its own for roughly 6,000 years before the first synthetic insecticide was ever compounded in a laboratory. Your grandparents did not purchase bottles of chemistry to manage their gardens because the relationship was already in place and already sufficient. The exchange between hunter and prey that Solano documented in July 2019 had been running, unassisted, since roughly 4000 BC. We simply interrupted it. What we replaced it with was an expensive, resistance-generating chemical system, and then we called the replacement progress. That reframing is the most consequential piece of marketing the agricultural industry ever produced, and most of us absorbed it as common sense. The economic comparison is not flattering to the chemical side. A €280 investment in 10,000 living predators outperformed €4,800 in 11 weeks of chemical management across the same crop area. Not because the natural option is inherently cheaper, but because it had already solved this problem long before anyone priced it. Now, you might be forming a doubt here, and it is a fair one. If the result was this clean, why are biological control programs still a minority practice across commercial agriculture in 2024? The answer is coming, and it lives in the economics of the replacement, not the biology of the solution. Stay with me. Solano's 2021 published report documented aphid density dropping to statistically negligible levels across all 6 treated rows within 72 hours of release, a result her own training data had not contained a reference point for, which tells you something important about how narrow the research baseline actually was. Recovery is not hope. Recovery is biology returning to its equilibrium once the pressure that displaced it is removed. That is not poetry. That is physics, and Daria Solano's field notes are the measurement. Drop your region and one species you have personally watched disappear from your area in your lifetime in the comments. The geographic pattern of what has been lost matters, and I want to read it. And yes or no in the comments. Have you ever seen something come back that you were certain was gone for good? Because if the answer is yes, I want to hear that story. Share this with someone who still believes the damage is permanent, because the evidence says otherwise. In the next video, I am going to show you what happened when 4 beaver families were reintroduced to a degraded peat catchment in the Scottish Highlands, a river system that had lost 80% of its water retention capacity across 60 years of drainage management. The Scottish Environment Protection Agency resisted the reintroduction for nearly a decade, citing flood risk to downstream farmland in 2012, and within 3 seasons the monitored catchment was retaining water at a rate that exceeded every engineered solution the agency had previously funded. The unresolved tension is this: the same water the beavers held upstream is the water the farms downstream had built their entire drainage infrastructure to remove, and both sides of that argument are, in their own terms, completely correct. I'm Dr. Raymond Finch. I'll see you in the field.